Hello students. As we know, biomolecules is one of the important chapters for NEET. Let's try to revise some of the important topics from this chapter. So let's start with proteins. In this video, we will understand some basics of proteins, right? So first question is, what are proteins? Proteins are basically made up of amino acids, right? These are polymers of amino acids. Amino acids are simple compounds, and if you take these small units, individual units, and join them to form a chain, then this chain, when properly folded in a specific manner, you get a 3D structure called protein. And proteins play diverse role in living organisms. So the basic unit of any protein is amino acid. There are 20 different types of amino acids found in proteins. Now in our body there are hundreds of amino acids, but out of these only 20 amino acids are found in the structure of proteins. These amino acids are joined by a very strong covalent bond called peptide bond. So if you take amino acids, let's say these are amino acids, and join them, the bond which is connecting the amino acids is actually the peptide bond, right? This entire chain with many amino acid residues connected by peptide bonds, right? This entire chain is called a polypeptide chain. This polypeptide chain, when like coiled and twisted in a specific manner, it will form a 3D structure called protein. So all the proteins are made up of basic unit called amino acids, and there are 20 types of amino acids found in proteins. These amino acids are joined by a very strong bond called peptide bond. Now the question is, what's the structure of amino acid, right? We'll start with the structure of amino acid. All the amino acids which are found in proteins are alpha amino acids. So we have an alpha carbon to which all the important functional groups are attached. Also, we can say these are actually the substituted methane, right? The basic formula of methane is CH4. We have carbon to which 4 hydrogen atoms are attached. Now imagine if we substitute some hydrogen atoms with different functional groups, like an amine group or an acidic group, right? Accordingly, you will have an amino acid. So if we have to draw very basic, like a skeleton structure of any amino acid, we have to take an alpha carbon. Carbon's valence is 4, so let's attach the specific groups to it. Amino acid, right? The word amino means there is an amine group attached to the alpha carbon. Next, acids लिखा है यहाँ पे, right? So acidic group, carboxylic acid group COOH is present. Third, we have the hydrogen. Now this part is constant for all the 20 amino acids. This is constant. Like if we have to draw all the 20 amino acid structure, you can simply copy this thing 20 times. Now the fourth group which is attached here to the alpha carbon, we write it as R group. This is the side chain. This is the side chain, and this side chain is variable, right? So this will vary from amino acid to amino acid. So simply you take R group, let's say the simplest possible R group which we can attach will be obviously hydrogen, right? There is nothing simpler than this. So draw this skeleton here, alpha carbon, amino group, then the COOH group, H, the constant part is done. R group, let's say we take the simplest, hydrogen, then we get our simplest amino acid, glycine. Now if you replace this with some other group, right, you will get a different amino acid. So in such a way, with different R groups, you will get all the 20 amino acids found in proteins. Also, depending on whether these amino acids are present in acidic solution or basic solution, right? What's the pH of solution? Depending on this, the charge on amino acid is also different. This amine group over here, right, instead of NH2 linked here to the alpha carbon, it can get protonated, right? A proton H plus will be added and you will get NH3 plus. So overall a positive charge develop ho jayega, right? Overall positive charge will be developed. And here this COOH can lose a proton and become COO minus. So this will give a negative charge, right? Depending on the pH, the charge on amino acid will change. So the structure of amino acid actually changes with change in pH of the solution. So simply if we have to look this like how the effect occurs, then we can see here if there is acidic pH, right? That means you are having a solution in which you are having the particular amino acid. This is the like basic structure of amino acid. This carbon here over here will be the alpha carbon, to which we have the H, COOH and the amine group and the R group. R group whatever it may be, right? It is variable. Let's focus on these two groups only here. So this amine group, as you can see, in acidic solution, that means you have high proton concentration in the solution, right? Acidic means protons will be more. Now, as you have these free protons in the solution, the NH2 will acquire the proton and become here NH3 plus. So, the overall charge here on the amino acid, as you can see, is positive, right? In acidic pH, what is the charge? Overall charge here positive because of this. Next, let's go here. Okay, what can you see here? Here, basic pH. So proton concentration will be low. Solution will have less protons. Okay, so as you can see here, amine group has lost the proton. Okay, amine group has lost the proton. Also, means instead of NH3 plus, here you are having NH2. And this COOH has also lost the proton and here you are having COO minus. So overall charge over here is negative. So in basic pH or alkaline pH, the charge, overall charge on amino acid is negative. But the structure here in between, right? So there will be a particular pH wherein the amine group exists as NH3 plus and here the acidic group exists as COO minus. So the plus and minus, positive and negative charges are balancing each other. That means this particular structure of amino acid is actually electrically neutral. This is electrically neutral and this form of amino acid, the electrically neutral form, will be existing at a particular pH, not necessarily pH 7. Okay, pH will not be 7. It may be less than 7 or more than 7, depends on the amino acid. Also, we have different types of amino acid, acidic amino acids, basic amino acids, right? R group will also contribute to this charge. But if we take simpler structure here, then we can see here in this case the charges are getting balanced, right? So at what particular pH this electrically neutral form will exist will be specific for that particular amino acid. So for some amino acids that value is less than 7, for some it is more than 7, right? So pH is not neutral but the charge is neutral. And this form, right, electrically neutral form of amino acid is itself called as Zwitter ion, right? This is only called as Zwitter ion, right? So charge will be zero. Now let's see what are the various amino acids, the 20 amino acids which are found in proteins. We will start with the simplest amino acid which is glycine, right? So every amino acid is given a unique 3-letter code and a single letter code. Okay, so I have mentioned this also over here. So glycine 3-letter code will be GLY and single letter code will be G, right? This is how we will also go through all the codes for every amino acid found in proteins. Glycine is the first amino acid. See the structure. Now this part, as you can see, this is actually the constant part, right? Here, this is the alpha carbon to which we have amine group which is protonated. Here COOH, it has lost a proton. So charges are balanced. So we have drawn the Zwitter ionic form. Then the third hydrogen part, this is the constant, right? This part which I have marked here is the constant. Like we will copy this 20 times for every amino acid. Now this here over here is the R group. And what's the R group here? H, right? The simplest possible thing which we can attach here, H. So this is the simplest amino acid, glycine, right? Very important point. The simplest amino acid is glycine. Next, alanine. And see the 3-letter and single letter codes also. Okay. Let's see the structure. This is the R group, CH3. So if methyl group is present as R group, then it will be alanine. So see, H, instead of H, here we are having CH3. So replace the R group, you will get different amino acid. The third, valine. See the 3-letter code, VAL and single letter V. See the structure, here we are having this as the R group. As you can see here, we have CH, then 2 methyl groups attached to it, right? This will be valine. Next, amino acid structure, as you can see here, leucine, right? Leucine is CH2, CH, and 2 methyl groups attached to this. This is the R group. And obviously this part over here, this is same, the basic copy paste matter, right? Same. This here over here will be the R group, right? This is leucine. Also, we have another amino acid, isoleucine. Okay, see the codes also. Isoleucine is ILE and the single letter code is I. Okay, and as you can see here, what's the R group? This one. We have CH to which a methyl group is attached, and then here you can see this CH2, CH3, methyl group, right? So far we have seen 5 amino acids out of 20. Glycine, alanine, valine, leucine and isoleucine, right? Now if you go through this, these are the basic structures and all these alanine, valine, leucine, isoleucine, they have aliphatic side chains, okay? Let's see further structures of amino acids, okay? The remaining 15 structures and also their natures. All of you have to know the nature of different amino acids. So here proline, okay, the 3-letter code is PRO, pro, and single letter is P. Now see the structure, this is quite different, right? Let's say this is a constant thing. And now see the side chain. Usually we have seen the side chain is attached only to this alpha carbon. Okay, all for all the other amino acids. But here we can see the side chain is attached to alpha carbon and also to the amine group over here. Right? So instead of amine, actually an imino group is formed. Now here I have drawn it in as a protonated, a charge form. But if you remove the charge, if you remove the proton, this will become NH. Right? Instead of NH2, it will be NH. So instead of amino, it is imino group. Okay? And see the side chain part, CH2, CH2, CH2. So simply write CH2, CH2, CH2. One end is connected to alpha carbon and other end is connected to the nitrogen over here, right? And instead of amine, you are having imino group. So this is the imino acid. And out of 20 amino acids, proline is the only amino acid, right? Proline is amino acid out of all the 20 amino acids. Next structure, we have lysine. See the codes also. And here, the single letter code is K. Now, we can't use L because we have already used the letter L for leucine, right? So, we have to use some other letter. So, we have used K. See the side chain. CH2, 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 CH2 and we have the amine group, protonated form, right? NH3+. We have an extra amine group, right? Now, we also have amine over here and we have this COOH or COO minus over here attached to the alpha carbons. Amine is basic COOH is acidic, so they were balancing each other. But now in the side chain, we are having an extra amine group. So overall nature of this amino acid will be basic. See, you can see two basic groups, one acidic group. So obviously overall the amino acid will be basic. So out of all the 20 amino acids, we can see there are 3 amino acids which are basic in nature. We have lysine, arginine and histidine. So one of which we have seen here right now, lysine. Next amino acid, this is also the basic one, arginine, and the code, single letter code over here is R, because we have already used the letter A for alanine, right? So arginine, see the side chain and just observe, can you see the amine groups over here in the side chain? As there are extra amine groups, what will happen? The overall nature of the amino acid will be basic. Next we have histidine, and see the structure. This will be the side chain part, right? It also has the amine groups, and that is why this is also basic amino acid, and there is a ring structure over here. Lysine, arginine and histidine, these are basic amino acids. Basic amino acids. Next, aspartic acid, okay, and see the single letter code D, again because we have used A for alanine, right? Simple. See the side chain. We have CH2, COOH, we have removed the proton part. So this is COO minus, CH2, COO minus, or simply CH2, COOH. As there is an extra carboxylic acid group in the side chain here. This carboxylic acid group is definitely there. This amine group is there, but they were balancing each other. But we have an extra acidic group. So overall nature of this amino acid will be acidic, right? Aspartic acid. Next we have glutamic acid, and the single letter code over here is E, because we have already used the letter G for glycine, right? So here glutamic acid is represented by E. So you can easily remember both these, aspartic acid and glutamic acid. Both are acidic. Structures are also very simple. CH2, CH2, and here there was COOH, right? So again, carboxylic acid group is present in the side chain. So overall, this will be an acidic amino acid. Both these, aspartic acid and glutamic acid, are acidic amino acids. Okay. Next, asparagine. Okay. See the 3-letter code ASN, and we have the single letter code which is L. See the structure. Now, if you compare aspartic acid and asparagine, there is only one difference, right? Instead of this O minus, or it was simply OH over there, we have removed proton, right? So instead of this OH, we have replaced it with amine group. So this becomes an amide group over here. What's this? Amide group, right? So this is amide containing amino acid. Okay? As you can see here CH2, COOH, and over here CH2, C double bond O and NH2. So the OH got replaced by NH2. All other stuff is as it is, right? So from aspartic acid, you can easily remember the structure of asparagine. In asparagine, we are having this amide group. Next, similarly, glutamic acid. If you know the structure of glutamic acid, you can know what is the structure of glutamine. The single letter code is Q and 3-letter code is GLN. Now see over here, we are having a side chain as CH2, CH2 and C double bond O NH2. So here also we have done the same thing. See the glutamic acid structure. Instead of this O minus or this OH, we have the amine group. We get the glutamine, right? These two, asparagine and glutamine, are amides. Okay, in the side chain, amide is formed, right? Amide group. Next, serine. Okay, 3-letter code SER and single letter S. Here the side chain, very easy, CH2 OH. So there is hydroxyl group here, OH group. Next, we have threonine. See the symbols THR and then single letter T. Here also you can see the side chain CH, and to it we have methyl and the OH group attached. So these two are OH group containing amino acids, right? Next we have methionine. See the codes also. And one different thing which you can see here is this has sulfur, right? CH2, CH2, S, CH3. So this is sulfur containing amino acid. There is one more sulfur containing amino acid which is cysteine. See the symbols also. As you can see, CH2SH. So there is sulfur over here, right? So out of 20 amino acids, there are two amino acids, methionine and cysteine. These have sulfur in their structure. Okay. So sulfur containing amino acids. Next, phenylalanine. Okay. The codes here, three letter code PHE and single letter code is F. Now we can't use P because P for proline, we have already used it. So phenylalanine is represented by the letter F. Now see here, what's the structure? This is the constant part and this is the side chain part, right? In alanine, our side chain was CH3. Remove one H and attach benzene ring, right? You can see aromatic benzene ring over here. So the side chain is CH2 and a benzene ring attached. This is phenylalanine, okay? And as you have this benzene ring over here, this is an aromatic amino acid. Next, tryptophan, right? See the symbols also. TRP is the three letter code and W is the single letter code. Again, we can't use T because we have already used it for threonine, right? See the side chain. This is the side chain part. CH2, then this 5-membered ring with again amine and benzene ring over here, 6-membered, right? Benzene ring. So aromatic. Again, as you are having a benzene ring, ये क्या है? Aromatic amino acid tryptophan. And last, tyrosine. TYR and the single letter code is Y, right? See the structure here also CH2, then a benzene ring, and we have OH, right? So if you know the structure of phenylalanine, simply attach OH here. You will get tyrosine. Simple, right? As here also you are having the aromatic benzene ring, tyrosine is also an aromatic amino acid. So these three, phenylalanine, tryptophan and tyrosine are aromatic amino acids. Very important point for your NEET examination. So now we have seen all the amino acids. So we will quickly go through the nature and all the 20 amino acids. Okay. So the simplest amino acid is glycine. Next, the other amino acids which have aliphatic side chains are alanine, valine, leucine and isoleucine. Next, aromatic amino acids which we saw on the previous slide, phenylalanine, tryptophan and tyrosine. Basic amino acids means they have extra amine groups in their side chains. So we have lysine, arginine and histidine. Acidic amino acids, these have COOH groups in their side chains. So aspartic acid and glutamic acid. Next, amide group containing amino acids. So amides, asparagine and glutamine. Sulfur containing amino acids are methionine and cysteine. Next, OH group containing we have serine and threonine. Amino acid, a very different structure, right? Proline. So this is how we have summarized all the 20 amino acids. Now in the next video lecture, we will understand how to connect these amino acids and form a polypeptide chain, and also how basically we can understand protein structure and all its levels in detail. That will be covered in next video lecture. Now, as we have completed basics about proteins and amino acids, I have few questions for you. Okay, so it's time for the knowledge drill. I will give you two questions and you have to tell me the answers to these questions in the comment section. Okay, so the first question for you. Name the bond which connects the amino acids to form a polypeptide chain. And second, you have to tell me the names of aromatic amino acids found in proteins. I hope this video was helpful for you. Thank you.